You know, every single thought you have, every memory you pull up, every tiny movement you make, it all comes down to a conversation. A microscopic, absolutely lightning-fast conversation happening between your brain cells. So today, we're going to listen in. We're going to decode that conversation and follow the incredible journey of a neural signal from one neuron to the next.
So here's our game plan. First, we'll look at the actual physical parts, the anatomy of the synapse. Then, we'll watch what happens when a signal arrives and triggers the release of a chemical message. After that, we'll meet the two main players, the brain's primary go signal, glutamate, and its stop signal, GABA. We'll see how those signals create tiny electrical changes, and then, for the grand finale, we'll see how a neuron puts it all together to make a decision.
All right, let's start at the beginning. Before you can understand the message, you've got to understand the mailroom, right? So we're going to start by exploring the physical stage where all this magic happens, the anatomy of the synapse. Getting a handle on the structure is absolutely the key to unlocking how it all works.
Okay, take a look at this. This diagram basically maps out the flow of information through a single neuron. The best way to think about it is like a relay race. The signal, the baton, is received right here in the input zone. That's the dendrites. It then travels toward the cell body. And if that signal is strong enough, it hits the trigger zone at the axon hillock. That's the starting pistol, firing off an action potential. That electrical signal then just zips down the conducting zone, this super long axon, all the way to the output zone at the terminals, where it's ready to pass that baton to the next runner, the next neuron in the chain.
Now let's zoom way in on that input zone. See, the dendrites aren't just smooth branches. They're actually covered in these thousands of tiny little protrusions called dendritic spines. You can think of these as the dedicated receiving docks, the primary mailboxes for pretty much all the incoming excitatory messages. They are absolutely essential for receiving signals, and as we're about to find out, they're anything but static. And wow, these images really give you a stunning look into that world. On the left, that's a real micrograph showing a dendrite that is just covered in these spines. And then on the right, you've got a 3D reconstruction that really shows off their complex, almost mushroom-like shapes. These aren't just simple bumps on a log. They're incredibly intricate, highly specialized little pieces of biological machinery.
So why are these little tiny spines such a huge deal? Well, for a few really incredible reasons. First, they are highly plastic. That means they're not fixed. They're constantly changing their shape and their size, and this is one of the fundamental ways we think learning and memory actually work. Second, each and every spine acts like its own little self-contained biochemical compartment, able to control its own internal calcium levels completely independently from its neighbors. Third, its exact shape makes a huge difference. Longer, skinnier spines have less of an electrical impact on the neuron than shorter, stubbier ones. And maybe the most amazing part? These spines are dynamically forming and disappearing all the time based on how much they're being stimulated. All of this taken together allows a neuron to fine-tune the impact of every single one of its thousands of inputs. It's just incredible. Okay, so our signal, our action potential, has raced all the way down the axon. Now it's reached the end of the line. But it faces a gap, the synapse. To get its message across to the next neuron, that purely electrical signal has to be converted into a chemical one. Let's dig into exactly how that amazing transformation happens.
It all happens in this beautiful, precise, three-step dance. First, the action potential arrives at the terminal. That change in voltage acts like a key, slamming open these special doors called voltage-gated calcium channels. Second, with the doors open, calcium ions flood into the terminal. This rush of calcium is the critical trigger. It tells these little bubbles, these synaptic vesicles that are filled with neurotransmitters, to move to the edge of the cell and fuse with the membrane. And third, that fusion event is like popping the bubble. It dumps the neurotransmitter out into the synaptic cleft, that tiny gap between the neurons. From there, the chemical message is free to float across and bind to receptors on the next cell, delivering the message.
So now that we know how the message gets sent, let's talk about what the message is. And the main messenger for get excited in the central nervous system is a molecule called glutamate. When glutamate is released, its basic message to the next neuron is, let's get more active, it's time to fire. It is without a doubt the brain's #1 go signal.
But here's the thing. Glutamate is a sophisticated messenger. It doesn't just have one way of sending its go signal. It uses two major classes of receptors. First up, you have the ionotropic receptors. The best way to think of these is like a key that fits directly into a lock that is the door. As soon as glutamate binds, the channel opens instantly and ions can flow through. This is all about speed, mediating super fast synaptic transmission. The second class is the metabotropic receptors. These are more like ringing a doorbell. Glutamate binds, but it doesn't open a channel itself. Instead, it triggers a chain reaction, a whole cascade of chemical signals inside the cell. This creates a much slower, more modulatory effect that can last a lot longer. Okay, let's zoom in on those fast-acting ionotropic receptors, starting with the AMPA receptors. These really are the workhorses of fast excitation in the brain. They're built from 4 protein subunits, and when glutamate binds, they pop open and primarily let positively charged sodium ions rush into the cell, causing that excitatory signal. But, and this is a really crucial detail, the exact combination of subunits matters. Most AMPA receptors contain a specific subunit called GluR2, and its job is to block calcium from getting through. But if a receptor is built without that GluR2 subunit, the channel suddenly becomes permeable to calcium too. And letting calcium in is a much more powerful signal that can trigger a whole host of other long-term changes inside that neuron.
Next up are the NMDA receptors, and these things are just fascinatingly clever. To open up and do their job, they have 2 special requirements that have to be met at the exact same time. First, they need glutamate to bind, sure, but they also need a little helper molecule, a coagonist, like glycine. So that's hurdle number 1. But even when both of those are bound, the channel is still physically plugged up by a magnesium ion. That's hurdle number 2. The only way to get this magnesium ion out of the way is if the neuron is already partially excited or depolarized by other inputs. This amazing dual requirement makes NMDA receptors brilliant coincidence detectors. They only send a signal when 2 things happen at once. Glutamate is present and the receiving neuron is already active.
And this slide shows that mechanism perfectly. Look at the image on the left. When the neuron is just sitting at its resting potential, you can see that magnesium ion sitting right in the channel pore. It's like a cork in a bottle. It physically blocks any ions from getting through, even if glutamate is bound. But now look at the right. When the cell becomes depolarized, when it gets more positive inside, that positive charge actually repels the positively charged magnesium ion, literally popping it out of the channel. This unblocks the pore and allows a flood of calcium to rush into the cell, carrying a really powerful signal for change.
So what about those slower metabotropic receptors, the doorbells? Well, they fall into 3 main groups. Group 1 receptors are generally excitatory. When they're activated, they kick off a chain reaction that ultimately leads to an increase of calcium inside the cell. Groups 2 and 3, on the other hand, are usually inhibitory. They both work by shutting down an enzyme that produces a really important internal signaling molecule called cyclic AMP, or CAMP. You can think of CAMP as an internal or second messenger. So by reducing the amount of this messenger, these receptors basically tell the cell to quiet down. This allows for much more subtle, nuanced, long-lasting control over how excitable that neuron is.
All right, for every go signal, the brain absolutely has to have a stop signal. It needs brakes. That critical counterbalance to glutamate's excitation is provided by a neurotransmitter called gamma-aminobutyric acid, or GABA for short. GABA is the principal inhibitory neurotransmitter. Its job is to tell neurons to calm down and become less likely to fire.
And just like glutamate, GABA has both fast, ionotropic, and slow metabotropic receptors to deliver its message. The GABA-A receptor is the fast one. It's a channel, and when GABA binds, it opens and allows negatively charged chloride ions to rush into the cell. Making the inside of the cell more negative is the classic, surefire way to cause fast inhibition. The GABA-B receptor is the slow one. It's a metabotropic receptor that works indirectly. It can either open up potassium channels, which lets positive charge leak out of the cell, or it can inhibit calcium channels to prevent positive charge from coming in. Either way you slice it, the result is the same, a slow, more prolonged inhibitory effect.
And you can see the difference in their design right here. On the left, that's the GABA-A receptor, a classic channel made of 5 subunits that sits right in the membrane, ready to open. On the right, you have the GABA-B receptor. It's actually a pair of proteins, a dimer, that snakes its way through the membrane and has to couple to other proteins inside the cell to do its job. 2 totally different structures built for 2 very different kinds of inhibition. Okay, so we've got neurotransmitters binding to receptors, and we've got ion channels opening and closing. But how does that chemical event actually translate back into an electrical signal in the next neuron? The answer to that is something called postsynaptic potentials.
This slide is so cool. It gives us a really remarkable look at the building blocks of one of these neural signals. This data comes from a technique called patch clamp, which is so sensitive it can actually measure the electrical current flowing through a single ion channel. Each one of these tiny little downward blips at the top represents one channel opening for a split second, letting in a minuscule current. But when a neurotransmitter gets released, it doesn't just open one channel, it opens thousands of them all at once. And all their tiny individual currents add up, they summate, to create the much bigger, smoother, measurable signal you see at the bottom. That's what we call an excitatory postsynaptic current, or an EPS.
So that flow of ions, that current, inevitably changes the neuron's voltage. And that change in voltage is what we call a potential. Now here is the absolute key takeaway. An excitatory current from glutamate creates an excitatory postsynaptic potential, or an EPSP. This is a little electrical nudge that brings the neuron closer to its firing threshold. On the flip side, an inhibitory current from GABA creates an inhibitory postsynaptic potential, or an IPSP. This is a nudge in the opposite direction, pushing the neuron further away from its firing threshold, making it less likely to fire.
And these potentials have some really distinct characteristics. For one, the size of an ESPS, its amplitude, is directly proportional to how much neurotransmitter was released. More glutamate means a bigger ESPS. Simple as that. We also see different timing. AMPA receptors give you these really fast, sharp ESPSs that last just a few milliseconds. NMDA receptors, once they get unblocked, contribute a slower, much longer-lasting tail to that signal. And it makes perfect sense. If you were to experimentally block the inhibitory IPSPS, if you cut the brake lines, you remove those stop signals, and the neuron becomes way more likely to fire action potential.
Now here's where it gets really mind-boggling. A single neuron isn't just getting one of these signals. It's getting thousands of these little EPSPs and IPSPs from other cells, all at the same time, all over its dendrites. This brings us to the final crucial step, integration. How in the world does the neuron add all this up to make a final fire or don't fire decision?
Well, the neuron is basically a tiny little calculator, and it uses two main types of addition. First, there's temporal summation. This is all about timing. It's what happens when multiple signals arrive from the exact same synapse, one after another, in really rapid succession. Think of it like multiple quick drum beats from one spot. Their little EPSPs add on top of each other. Second, there's spatial summation. This is all about location. It's what happens when signals arrive at different synapses all across the neuron at the same time. The neuron adds up all these inputs from different places. And at the end of the day, that's what a neuron is doing, constantly. It's summing all the excitatory go signals and subtracting all the inhibitory stop signals. If the grand total, when it reaches the axon hillock, is enough to push the membrane potential past that critical threshold, boom, an action potential is fired. A decision is made.
And that really brings us to the final, awe-inspiring thought. We've gone all the way from the physical shape of a synapse to the release of chemicals to these tiny little electrical potentials, and it all leads to this, a constant dynamic process of summation. So the question I want to leave you with is this. How does this simple arithmetic, this relentless, ongoing addition and subtraction of go and stop signals, happening trillions of times every single second in your brain, somehow give rise to the incredible complexity of everything you perceive, everything you think, and everything you do?